Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the oxygen dissociation curve and then finally we studied about the transport of oxygen. And for the oxygen what we are saying 97% of the oxygen is transported through the hemoglobin present in the RBC. Yes or no? So that is what the content that we are saying here. So here this oxygen is attached to the hemoglobin and hemoglobin is present generally as a tetramer. Generally it is present as a tetramer. So though we are writing like this, originally it is Hb4. It is a tetramer plus, and here each one is binding with one oxygen. So since it is a tetramer, overall it is binding with 4 oxygen molecules. So that's how it is forming Hb4O8. This is what the technical one, but generally at the level of this 11th class you don't need to worry about this much of more details. But please remember, overall here it is binding with 4 oxygen molecules or 8 oxygen atoms, and this attachment is not causing oxidation of Fe+2. So that's why it is not oxidation process, simply oxygen is attached, that's why it is considered as oxygenation process. Yes or no? At the alveoli, due to high partial pressure, now oxygen is binding with the hemoglobin and this oxyhemoglobin is transported to the tissues. And at the tissues what happens, this oxygen will be dissociated so that it will be going into the tissues. Yes or no? So this is how oxygen is transported. And apart from this, approximately 3% of oxygen is directly dissolved in plasma. So in the dissolved form it can be transported to the tissues. So this is what the oxygen transport. So it is a very simple one. But now we are focusing about carbon dioxide transport. So transport of carbon dioxide, and this carbon dioxide is produced within the tissues. So in that cellular respiration we studied, now oxygen is utilized and CO2 is released. Now at the tissues this CO2 is entering into the blood, and from there it should reach the lungs, means alveoli. So how exactly it is coming from the tissues to the alveoli? Through this blood. So here for the CO2, in the previous lecture also we studied about this. So the CO2 is transported in 3 different ways. 7% is dissolved in the plasma. 7% is dissolved in plasma. So here what happens, directly in the blood plasma it is dissolved. So along with the blood plasma it is reaching the alveoli. That's it. So there is no much explanation here. But the point that we need to remember is solubility of CO2 is much higher than the solubility of oxygen. Already we studied about this. So around 20 to 25% more CO2 is soluble when compared with oxygen. Okay. So directly in the dissolved form it is reaching this alveoli. Over. For this is completed. But the next two are very important. Around 20 to 25% of CO2 is transported as carbamino hemoglobin. Okay, not carboxy. Many students are making mistake here. Carbamino hemoglobin, not carboxy. When carboxy hemoglobin will be formed, if carbon monoxide, if CO is attached with hemoglobin, then it will form carboxy hemoglobin. But this is CO2 that is attached to hemoglobin, so as carbamino hemoglobin. I will tell you details. And around 70% CO2 is transported as bicarbonates. Sodium bicarbonate, potassium bicarbonate. So in the form of bicarbonate it is transported. Now anyhow, this is directly dissolved in plasma. So for this we don't need to study separately. But for these two we need to observe very clearly. So the mechanism is very simple, and finally we will read the NCERT content. Now let us observe carefully here. Now let us talk about the tissues here. So it is having the cells. Okay, so this is at the tissue level. At the tissue level, what is happening? Oxygen is utilized. Suppose I am drawing this blood vessel. So observe carefully here. Suppose this is the blood vessel. Okay, and in this blood vessel, now I am making circle here because we need to use the space effectively. So that's why I am using like this. And here we can see the lungs. So you please imagine that this is the blood circulation. Okay, so from the lungs blood is coming to the tissues, from the tissues again it is going to the lungs. So what exactly happening at the tissues, what exactly happening at the lungs? These are the points that we need to observe very clearly. So observe this. tissues are producing CO2 and releasing CO2 into the blood. Yes or no? So they are releasing the CO2 into the blood. From the out of this CO2, 7% is dissolved in the plasma. 7% of CO2 is directly dissolved in the plasma. So in the dissolved form only it is reaching the lungs. In the dissolved form only it is reaching the lungs. And this can go into the lungs. Yes or no? This is what they are saying here. And then what about the remaining? So 7% means out of 100 remove the 7. That means around it will be 93%. 93%. Out of this 93%, 70% as bicarbonate. 20 to 25 they gave. So here you can get 23% as carbaminohemoglobin. So now we are focusing about this 93%. For this 93% we are talking now. So please observe carefully. And what happens to this 93%? This 93% CO2 is now entering into RBC. Is entering into RBC. This is the RBC. Okay. So red blood corpuscles are red blood cells. So into that they are entering. Now inside this what happens? Now once this CO2 entering into this RBC. Now what happens? So regarding the 70% we are talking now. This CO2 will combine with water. CO2 will combine with water and it will form H2CO3 carbonic acid. H2CO3 carbonic acid. And immediately it will dissociate as H plus and HCO3 minus. It is dissociating as H plus and HCO3 minus. Is it clear? Now this HCO3 minus, now it is formed inside the RBC. And now this HCO3 is coming into plasma. HCO3 minus is coming into the plasma. So since one negative ion is coming into the plasma, one negative ion should enter into the cell, then only the charge will be maintained. So who is entering into this particular one? Now observe carefully here. So who is entering here? From the plasma, Cl minus chloride ion is entering into the cell. So observe carefully here. HCO3 minus is coming out of the cell, and to compensate that charge loss, now Cl minus is entering into the cell. So in this way, HCO3 minus is coming out of the RBC, Cl minus is entering into this, and this particular step is called as chloride shift. It is called as chloride shift, and you can also call this as Hamburger's phenomenon. Hamburger's phenomena. This Hamburger's phenomena is nothing but chloride shift. Since one negative charge, that negative ion, that is HCO3 minus, is coming out of RBC, Cl minus, that is also one single negative that is going into the cell to balance the charge across the cell. So that is the reason why this is called as chloride shift. It is also called as Hamburger's phenomena. Clear? And now this HCO3 minus, can combine with Na plus. So that now it will form sodium bicarbonate, NaHCO3. Yes or no? Suppose it is combining with potassium, it will form KHCO3, potassium bicarbonate. So in this way, in the form of bicarbonates, 70% of CO2 is transported. This is what they are saying. I am telling you in simple, so please try to understand this. Anyhow, out of the total CO2, approximately 7% is dissolved in plasma and it is going directly. And remaining CO2 is entering into the RBC. And out of this, approximately 70% CO2 combining with water. And here there is an enzyme that is called as carbonic anhydrase. Regarding this reaction, already we studied in biomolecules. So please remember this. CO2 plus H2O in presence of carbonic anhydrase enzyme, it is forming carbonic acid. And this carbonic acid is immediately broken down into H plus and HCO3 minus. That HCO3 minus is coming into the blood plasma, so that Cl minus is entering into RBC. And this particular step is called chloride shift or Hamburger's phenomena. And now this bicarbonate HCO3 minus may combine with Na plus or K plus, so that it is forming the corresponding bicarbonates. So now these bicarbonates are going to the lungs. Understood or not? This is what. Then what about the remaining CO2? And some amount of CO2 is combining with hemoglobin, but please remember this hemoglobin is combining with this H plus. So that's why how exactly we are writing is, we are writing this as HHB, observe carefully here. So this is written as HHB. So we are writing this as HHB because this H plus is combining with hemoglobin. So this HHB, now this HHB with CO2. Now this is forming this HHB CO2. You can simply write it. You can simply write like this. Okay. So this is how we are writing like this. Understood or not? For your understanding. And now this is called as carbamino. So that's why it will be generally written as HHB, and here it will be attached to NH group, and here there is a rearrangement, so COOH. So in this way it will be attached. This is the carbamino hemoglobin. Understood or not? This is how it will be transported. It is going here itself like this as it is. So in this way total 3 mechanisms are there. One is dissolved form, that is the directly it is dissolving in the blood plasma. Other one is in the form of bicarbonates, and the other one is carbamino hemoglobin. In this way, now this blood, and the RBC means it is not present here. It is also going along with the blood flow. RBCs are also going to the lungs. Now at the lungs what happens? Exactly opposite reactions will occur. What is the meaning of this exactly opposite reactions here? Exactly opposite reaction means, see here, this Na plus NaHCO3 is there. It is divided as, separated as Na plus and HCO3 minus. So that now HCO3 minus will enter into the RBC. HCO3 minus. If HCO3 minus is entering into the RBC, Cl minus will come out of this, and now this is called as a reverse chloride shift. It is called as a reverse chloride shift. In the chloride shift, what is happening? Cl minus is entering into RBC, HCO3 minus is coming out. But here it is opposite. HCO3 minus is entering into RBC, Cl minus is coming out. That's why it is called as reverse chloride shift. So chloride shift is occurring near the tissues. Reverse chloride shift is occurring at the tissues. Clear. Now HCO3 minus entered here. Now here what happens? This HHbCO2. From this hemoglobin also CO2 is coming out. From the hemoglobin also CO2 is coming out. And then from the hemoglobin H plus is also coming out. So that H plus will be removed. That H plus is removed. Now hemoglobin is present separately. Yes or no? Now from the lungs oxygen will come to the hemoglobin, oxygen will attach. That is called oxyhemoglobin. Now from this HHB, now H plus is released. Now this H plus will combine with and it will form H2CO3. And again the reverse reaction will occur and it will form H2O plus CO2. And this CO2 will come into this blood, and now again it is going into the lungs. Understood or not? This is what happening. That means whatever is happening here, exactly opposite is happening here. That's why at the tissues, if you understand clearly, exactly opposite you can write very easily. Understood or not? Once again I will repeat here. So at the tissues what is happening? Within the RBC, CO2 combines with water and it is forming the carbonic acid in presence of carbonic anhydrase, and it is broken down as H plus and HCO3 minus. HCO3 minus is coming into the blood plasma, combining with some sodium or potassium and forming corresponding bicarbonates. Now in this way it is going there up to the lungs. Next one here, the released H plus is generally combining with this hemoglobin, and there it is also combining with some amount of CO2. Approximately 20 to 25% CO2 is attached. So finally it is forming the carbamino hemoglobin. I will write here. Carbamino hemoglobin. Why it is called amino? Please understand this. In the case of hemoglobin structure, I am randomly writing this. There are 2 alpha chains, 2 beta chains. And what are these chains? They are actually some proteins. They are amino acids. They are having amino acids. Amino acid is having amino group here. Amino group here. Yes or no? And here to the amino group of these particular chains, now this CO2 is attached. Now you just observe carefully here. Here also 4 CO2 molecules are attached to hemoglobin. 4 CO2 molecules are attached to hemoglobin. So please observe this very carefully. So one hemoglobin can combine with 4 CO2 molecules. That is what the logic is, and that's why it is called as carbaminohemoglobin. So in this way, bicarbonate and carbaminohemoglobin, and then dissolved form of the CO2. These are the ones we are studying at the tissues. Now they are going to the lungs. Lungs means what? Alveoli are present. Alveoli are supplied with some capillaries. This is what in the capillaries means, it is a blood circulation. At the capillaries, what happens? This dissolved form directly as it is, it can directly diffuse into the lungs. But regarding this bicarbonate, they are present in the blood plasma. Now it is again dissociated here. Now bicarbonate ion is formed. Now it is going into the RBC. So that Cl minus is coming out of RBC to balance the charge. And now this HCO3 minus is combining with H plus released from this hemoglobin. H plus released from the hemoglobin. So that it is forming carbonic acid. And again it is dissociated into H2O and CO2. Now this CO2 will come into the plasma and it will diffuse into the lungs. Means alveoli. Next one. Regarding the hemoglobin. To this hemoglobin here CO2 is attached. It is coming up to here. And here now more and more oxygen is coming from the lungs. Please remember. So this oxygen, when combined with this hemoglobin, it is forming oxyhemoglobin, that is acting as an acidic, that is acting as a acid. And there, in presence of this oxyhemoglobin, when more and more oxygen is present, now this CO2 will be easily dissociated. When CO2 is dissociated here, now it again diffused into the lungs. This is what? Very simple mechanism. Very simple mechanism. You don't need to get any confusion here. Very simple. 3 forms. All the 3 forms are very simple. So with this, in this way, from the tissue, CO2 is transported to the lungs or alveoli. So that there it is going into the lungs. During expiration, this CO2 is released into the atmosphere. That's it. Understood or not? Now we will see overall mechanism here in simple for both oxygen and CO2. So here we are talking about the lungs, and here we are talking about the tissues. This is the tissue. What actually we are saying here? Hemoglobin is present in the RBC here. Hemoglobin is present in the RBC. From the lungs, oxygen is coming and attaching with oxygen, attaching with hemoglobin. From the lungs, oxygen is coming into the blood circulation, and this oxygen is binding to hemoglobin and forming oxyhemoglobin. And 3% of oxygen is dissolved in the blood plasma. And now this oxygenated blood is coming to the tissues. Yes or no? At the tissues, what happens? CO2 levels are very high. Already we studied the factors. CO2 levels are very high and H plus ion concentration is more. H plus ion concentration more means pH value is less, and the temperature is also high. And 2,3 bisphosphoglyceric acid or 2,3 bisphosphoglycerate levels are high. And these factors, they are helping in dissociation of oxygen. Oxygen is dissociated from hemoglobin. Once oxygen is dissociated from hemoglobin, now it is released from hemoglobin. It is coming into the blood and then it is coming into the tissues. In this way, oxygen is going into the tissues. Yes or no? This is what the concept we are studying, right? So now, once oxygen is removed, hemoglobin will be free. Now this CO2, 7% of CO2 is dissolved in the blood plasma. Remaining CO2 is entering into the RBC, and some amount is converted into bicarbonate, and some CO2 is attached with hemoglobin. This is what. So here oxygen is removed. That means hemoglobin is becoming free. Now CO2 will attach to that hemoglobin. That's what happening. And now this is coming to the lungs. At the lungs, what happened? This CO2 from dissolved can directly diffuse into the lungs. But here bicarbonate entering into RBC, and finally it is releasing the CO2 that is diffused into the lungs. And the CO2 attached to hemoglobin, that is also released and it is also diffusing into the lungs. This is what the overall idea regarding the transport of both oxygen and the CO2. The forms, 3 forms for the CO2, 2 forms for the oxygen. How exactly they are going, that you need to understand. Clear? And with this transport of this oxygen is CO2 is also completed. But here we are using 2 names. At the level of tissues, we are using this word called as Bohr's effect. At the level of tissues, we are talking about the Bohr's effect. And at the level of the lungs, we are talking about Haldane's effect. Haldane's effect. What are these things? You can understand this very easily. What this Bohr's effect indicates? Bohr effect says that at the level of tissues, CO2 levels are more. When CO2 levels are more, now it is indirectly helping in dissociation of oxygen. Dissociation of oxygen. This is what? Dissociation of oxygen from hemoglobin, so that oxygen can easily enter into the tissues. And this particular phenomena is called as Bohr's effect. So in the case of Bohr's effect, if you are observing the Bohr's effect, suppose this is the standard curve. So where you can observe the Bohr effect? On the right side shift. Right side shift will indicate Bohr's effect. Dissociation, right? Dissociation. This is what? Bohr's effect at the tissues. So based on the Bohr's effect, you can say that in the conditions related to tissues, you can see that P50 value is increased. That is the Bohr effect. So Bohr effect is related to tissues condition where oxygen is easily removed from the hemoglobin and it can easily go into the tissues. Then what is this Haldane's effect? Haldane's effect is opposite. Haldane's effect we are studying at the lungs. At the lungs, what you are saying, this oxygen from the lungs is going into this RBC, so that now it is causing the separation of CO2 from hemoglobin, so that this CO2 can easily go into the lungs. And not only this, there are some factors here. What are the factors here? High partial pressure of oxygen, low partial pressure of CO2, and then low temperature, and then low H plus ion concentration, that means more pH value, and then low 2,3-bis-PGA levels. And these are the factors that we are observing at the lungs, so that these factors will help in easy diffusion of CO2 into the lungs. That's it. So in this way, we are talking about total 3 scientist names. So sorry, 2 scientist names. One is Bohr's effect at the tissues, other one is Haldane's effect at the lungs. Along with this, we are studying about the Hamburger's phenomena, and this is the chloride shift that we are studying at the tissues. Reverse is happening at the lungs, but it is called as reverse chloride shift. That's it. These are the various important names that you need to understand with respect to this transport of carbon dioxide. Concept is very simple. I hope you understood this. And now let us quickly go through this particular PowerPoint. Transport of carbon dioxide. 7% as dissolved form in plasma, 70% as bicarbonate, 22 to 25% as carbaminohemoglobin. Clear? And regarding the carbaminohemoglobin, see here, for each of these protein, amino group is there. To each of these amino groups, CO2 is attached. So in this way, one hemoglobin can carry 4 CO2 molecules. Clear? Next one. Next one here, this is whatever the reactions we have studied. Same reaction at the tissues, at the lungs they are talking. At the tissues, whatever we are studying, exactly reverse will happen at the lungs. Simple logic. Okay, this is what they are saying. Now let us go through NCERT content. CO2 is carried by hemoglobin as carbamino. It is not carboxy. Many students are making mistake. It is carried as carbaminohemoglobin. How much it is? About 20 to 25%. And this binding is related to partial pressure of CO2. So at the level of tissues, more partial pressure of CO2 is there. That's why CO2 can easily bind with hemoglobin. That's what they are saying. Sorry. PO2 means partial pressure of oxygen is a major factor which could affect this binding. So at the level of tissues, partial pressure of oxygen is lesser. So that's why it is helping in attachment of CO2 with hemoglobin. Suppose you are talking at the level of alveoli, what happens? Their oxygen partial pressure is more. If oxygen partial pressure is more, CO2 cannot easily bind with hemoglobin. That's why what they are saying, the binding of CO2 with hemoglobin is dependent on partial pressure of CO2 and also partial pressure of oxygen. So for the attachment, partial pressure of CO2 should be high and partial pressure of oxygen should be low. This is what the meaning is. So partial pressure of oxygen is a major factor that could affect this binding, because at the level of alveoli, oxygen partial pressure is more, then CO2 is not binding with hemoglobin, it is detached. That's what they are saying here. Please understand the sentence and what is its internal meaning. If you don't know internal meaning, there is no use in studying this. So please try to understand what exactly they are saying. Next one, when partial pressure of CO2 is high and partial pressure of oxygen is low, that where we are observing this? At the tissues. So when we are observing these at the tissues, more binding of carbon dioxide occurs. That's why at the level of tissues, more CO2 is binding with hemoglobin. Whereas when the partial pressure of CO2 is low and partial pressure of oxygen is high, where we are observing this? In the alveoli. Then what we are observing? So dissociation of CO2 from the carbaminohemoglobin takes place. That means at the level of alveoli what happens? CO2 is detached, dissociated from hemoglobin. That is CO2 which is bound to hemoglobin from the tissues is delivered at the alveoli. That means at the level of tissue, CO2 is attached to hemoglobin, and until the alveoli it is carried along with hemoglobin. But at the alveoli, now the CO2 is detached from hemoglobin so that it can easily go into the alveoli. That's what they are saying. Next one, RBCs contain a very high concentration of the enzyme. What is the enzyme? Carbonic anhydrase. Now the second method they are saying carbonic anhydrase, and minute quantities of same is present in plasma also. So that means what? Carbonic anhydrase in the large quantity present in the within the RBC, but a small quantity of this carbonic anhydrase is even present within the blood plasma also. That means a small amount of bicarbonate is formed even in the blood plasma also. That is what the meaning is. Please understand the meaning also. Next one, this enzyme facilitates the following reaction in both the direction. It is a double-headed arrow. It is a double-headed arrow. The reaction can occur in both the directions, so it is bidirectional. Please observe this carefully. So CO2 plus H2O in presence of carbonic anhydrase, it is converted into H2CO3. And the same enzyme is also catalyzing the separation of H plus from this, so that bicarbonate ion and then H plus ion proton are released. And this where we are observing this left to right side reaction we are observing at the level of tissues, and right to left side reaction we are observing at the level of alveoli. In both the cases the same enzyme, same reactions are occurring here. So it is a double sided reaction, it is a bidirectional reaction. Observe carefully here. So in this way, at the level of tissues, bicarbonate is produced and H plus is produced. And at the level of alveoli, reverse, CO2 and water are produced, and this CO2 will easily escape into the alveoli. This is what the concept they are saying. Next one, at the tissue site where partial pressure of CO2 is high due to catabolism. So catabolism, due to the breakdown of the various substances in the tissues, more CO2 is released, and this CO2 diffuses into the blood. So it is going into the blood plasma, and also it is going into the RBCs, and it is forming the bicarbonate and also H plus ion, because the enzyme is present in both RBC and plasma. So same reactions are occurring in both the locations. Okay, next one, at the alveolar site where partial pressure of CO2 is low. At the alveolus means in the lungs, what happens? The partial pressure of CO2 is very less, and the reaction is proceeding in the opposite direction, exactly opposite direction. So that here what happens? Opposite direction means what is formed? CO2 and water are formed. So leading to the formation of carbon dioxide and water. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli and is released as CO2. So at the level of tissues, CO2 is trapped or converted in the form of bicarbonate. Now in that form it is coming to the alveoli. At the alveoli again it is converted into CO2 and then it is escaping into the alveoli. That's what they are saying here. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to alveoli. Please understand this. For the oxygenated blood, what we are saying, oxygenated blood delivers 5 ml of oxygen. 100 ml of blood delivers 5 ml of oxygen to the tissues. That's what we studied. Now we are studying about CO2. Every 100 ml of blood, every 100 ml of deoxygenated blood delivers 4 ml of CO2 into the alveoli or the lungs. Now you need to understand the solubility logic here. What is the significance of solubility here? Please observe carefully here. So here we are saying that solubility for the CO2 is much higher. That is the reason why at the level of alveoli, if you are observing, at the level of alveoli, if you are observing. Now let us observe carefully here. So this is at the level of alveoli, and now we will see at the level of tissues. Now this is at the level of tissues. For the oxygen, we are saying that from the oxygenated blood, it is going into the tissues. In the oxygenated blood, partial pressure of oxygen is 95 mm mercury. 95 mm mercury. And in the tissues, it is 40 mm mercury for the oxygen. Now you just see huge pressure difference is there. Huge difference in the partial pressure is present, but still it is delivering 5 ml oxygen. Still it is delivering 5 ml oxygen because solubility of oxygen is lesser. You can understand this. Now at the level of this one, in the deoxygenated blood, what is the partial pressure of CO2? It is a 45 mm mercury. It is 45 mm mercury. But in the alveoli it is 40 mm mercury. In the alveoli it is 40 mm mercury. See the pressure difference. It is just a 5 mm difference. but still, how much of oxygen is delivered here? Sorry, how much of CO2 is delivered here? 4 ml of CO2 is delivered. You need to understand this. So that's why generally average values, if you are talking, so 100 ml of deoxygenated blood carries or transports. Generally it is carrying 50 ml of CO2. Generally it is carrying 50 ml of CO2. Out of this, in the normal condition, it is releasing 4 ml of CO2 into this alveoli. Please understand this particular point. The point you need to understand is this one. Even though huge pressure difference is there, here it is only 5 ml. Even the very less pressure difference is there, still it is delivering 4 ml. How this is possible? This is possible because of solubility. Solubility for the CO2 is much higher. If it is more soluble, it can be moving very quickly. So diffusion will be very fast. So that's why even for the less pressure difference also more comparatively with this. Now here it is releasing the more CO2 per unit difference in the pressure. This is what we are talking. This is what the basic important concept that we need to understand. The same point they are saying here. And next one here. So for every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. So whether it is transporting or carrying, and the other word is delivery. Please try to understand the difference in these words. Transporting or carrying are same, but delivering is something different. So it is giving, that is the delivery. But carrying means it is present within it. So this is how generally we are doing these calculations. Clear? Now Bohr's effect generally we are talking about at the level of tissues. It is regarding in respect to or with respect to oxygen release into the tissues. Whereas Aldane effect we are talking at the level of the lungs or alveoli, where how easily CO2 is going into the lungs. With respect to this we are talking here. And the Hamburger's phenomenon is also called as chloride shift we are observing at the level of tissues. Opposite to this we are observing at the level of alveoli or the lungs. This is what the information is. This concept is very simple. Don't try to by heart this. Try to understand the mechanism so you can easily understand this particular transport of the carbon dioxide. Clear? With this particular lecture is completed. So in the next lecture we will go with regulation and also means breathing, regulation of the breathing, and also some diseases names they have given, that also we will go through, and summary also quickly I will read. So these are the things that we are studying in the next lecture. Okay, thank you so much my dear friends. We will meet in the next lecture. Thank you.